Hi, I'm Sam Ozminkowski, and I'm going to be talking about identifying microbial drivers and biological phenotypes with the Bayesian Network Regression model, work I've done in collaboration with Professor Claudia Solis-Lemus. I'm going to start by briefly talking about why we're working on this approach, and then get into some high-level information about the model. The bulk of this talk will focus on simulation studies we've performed to evaluate the model, and then I'll touch on what comes next. So, we know that biologists want to study the relationship between microbiome networks and relevant responses. For example, in this 2019 paper, a study was performed to determine the relationship between microbial community complexity and ecosystem productivity. Here, the linkage density was used as a predictor, and various functions, such as nitrogen leaching, were used as outcomes. In humans, this 2010 study aimed to determine the relationship between human gut microbiome and Crohn's disease, comparing the abundances of 12 specific OTUs between Crohn's and healthy patients. This figure on this slide comes from the 2019 paper by Wag and colleagues. Section A shows bacterial richness, their predictor, under a few different conditions. Section C gives the values of some of the different outcomes studied, with nitrogen leaching on the far right, and Section B gives the microbial network under the first condition, that is, the condition given in the blue bar in sections A and C. So, we can see this as an active area of research for plant biologists as well as in medicine. This is a very active area of research in statistics as well, with a lot of interesting methods having been developed so far. Current methods can generally be broken down into one of a few different types. Some focus on the relationship between a single microbial OTU and the phenotype, but this procedure has assumptions which can be violated by the complexity of the microbiome, which can lead to elevated type 1 error rates and reduced power. For example, if a response was loosely related to the OTU under study, but the OTU under study and the response were both strongly related to a separate OTU not being studied, that could produce some adverse results. High-dimensional regression models solve this problem by allowing inclusion of multiple OTUs, but they can be complicated by problems such as multicollinearity or instability due to overfitting. Furthermore, these standard regression analyses rarely account for the interactions among microbes. And that's where Bayesian network regression comes in. This method was originally developed on brain connectome data by Guha and Rodriguez in 2020. In the original implementation, they aimed to determine influential brain regions and the connections between them on a creativity index. This model was very well performing in this application. So the figure here shows some of their results. Each cell in the matrix refers to a connection between two brain regions, and connections which they found to be influential are colored white. It's important to note, however, that brain connectome data is completely dense. Everyone has the same brain regions, and every sample has the same brain regions in it. And that's where our project begins. We wanted to evaluate the performance of this model on microbiome data. We aim to identify influential microbes and connections between them in some relevant response, generally plant or human. Unlike the brain application, microbiome networks are very sparse. Most microbes are not in most samples. We've also implemented the package for running this analysis in the Julia programming language, which is fast, easy to use, well documented, and currently available. So let's get into a little bit more detail about the model. In general, we view the response Y as the inner product of A, an adjacency matrix describing the relationship between the microbes, and B, a matrix of coefficients on those edges, plus some random error. This model allows us to get estimates not only of the coefficient matrix, but also of the probability that each microbe is influential on the response. And we obtain these estimates using Gibbs sampling. Now to get into the simulation studies. We split our simulations into two types, which we're calling unrealistic and realistic. Our unrealistic simulations identify how the model performs when the response is generated directly according to the model. We begin by simulating a 30-taxon phylogenetic tree and a true coefficient matrix. The phylogenetic tree is simulated using the R package APE. For the coefficient matrix, we first determine if each microbe is influential on the response by drawing from a Bernoulli distribution with either 0.3 or 0.8 as the probability that each node is influential. Then, we set each edge coefficient as follows. For every pair of influential microbes, we draw from a normal distribution with mean 0.8 or 1.6 and variance 1. For edges where one or both microbes are not influential, we set the edge coefficient to 0. Using two different values for the mean of the distribution allows us to test how the results of this model differ when the magnitude of the effect each edge has on the response differs. For each of 100 or 500 samples, we randomly select 8, 15, or 22 microbes from the phylogenetic tree. We generate the adjacency matrix for each sample using the inverse of the phylogenetic distances between each pair of microbes in the sample. This way, microbes which are more closely related to each other have higher values in the adjacency matrix. If a microbe is not in the sample, any cells in the adjacency matrix corresponding to that microbe have a value of 0. Finally, we calculate each response as the Frobenius inner product of the adjacency matrix and the true coefficient matrix, plus some random error drawn from the standard normal distribution. So let's get into some results. Here we show the estimated probability of influence for the microbes under various conditions. Note that K here refers to the number of microbes per sample, pi is the probability that each node is influential, and mu is the mean of the normal distribution the edge coefficients are drawn from. As you can see, using 0.5 as a naive cutoff, the model does a very good job determining which microbes are influential and which are not, with only 2 false negatives in the case with the sparse coefficient matrix, sparse adjacency matrices, and weak edge coefficients. That's the bottom left panel in the left plot. Additionally, the truly influential microbes tend to have probabilities of influence that well exceed 0.5, and the non-influential microbes tend to have probabilities of influence which are far lower than 0.5. Here, we have 95% credible intervals for the coefficients on the connections between microbes. For both of these plots, each sample has 8 microbes, with 500 samples total. Both show dense coefficient matrices. The plot on the left is for weak edge coefficients, with the plot on the right for the strong edge coefficients case. These plots are split so truly influential edges are in the top area and truly non-influential edges are in the bottom. Edges found to be influential by the model, defined as having a 95% credible intervals that do not include 0, are shown in dark blue. Finding influential edges, the model does slightly worse but still performs very well overall, especially in the case with strong edge effects. There are very, very few false positives, and in the strong edge effect case, not many false negatives either. Now for the realistic simulations, things are a little bit more complex. We again start by simulating a 30 taxon phylogenetic tree and an edge coefficient matrix, this time with mean 0.4. In addition to generating a coefficient matrix, we generate main effects for the presence of microbes. We do this in 2 different ways. For what we're calling the phylogenetic simulations, we draw from a normal distribution where the covariance matrix is determined by the phylogenetic tree. For the random simulations, the main effect coefficients are drawn from independent normal distributions. For both types, we use 0.8 or 1.6 for the mean of the main effect distributions. We then generate the response variables in 3 different ways. First, for the additive model, we only take into account the main effects. Again, we use a Bernoulli random variable to determine if each microbe is influential on the response, with the same probabilities as the unrealistic case, 0.3 or 0.8. This time we generate 500 or 1,000 samples. For each sample, we randomly select 8 or 22 microbes, and if a microbe is in the sample and influential on the response, we add its main effect coefficient to the response for that sample. For the interaction model, we bring in edge coefficients, keeping the main effects. So here the response is the sum of the coefficients on the influential microbes included in the response, as well as the coefficient on the edges between each pair of influential microbes. Finally, we have a functional redundancy model, where we use the same formula as the interaction model, but we cap the response based on main effect and coefficient matrix sparsity. For the interaction simulations, the model again performs well. Here, results are given for both sample sizes, with dense adjacency matrices. With 1,000 samples, the model does better separating influential from non-influential microbes, but the results look good with both sample sizes. For the edge coefficients, both plots here are for 1,000 samples with strong main effects. We see very few false positives, again, but this time there are slightly more false negatives, as opposed to the unrealistic simulations. Again, though, the model performs pretty well overall in this case. For the additive simulations, the model does not perform as well, especially with the smaller sample size. At a larger sample size, dense coefficient matrices and strong mean effects, it seems to do okay, but this is the best case scenario for this model. We can still see, however, that the model produces few false positives for the edge coefficients, though again, slightly worse than in the unrealistic case. So overall, with the unrealistic simulations, we see little to no effective sparsity, either in the coefficient matrix or the adjacency matrix. We see the edge effect is important, especially with small sample sizes. For the realistic simulations, sample size is very important. With the interaction and redundancy models, we see relatively good performance at high sample size. With the additive models, we see very poor performance in all cases except, again, that dense adjacency matrix at high sample sizes, which still shows relatively poor performance. As noted, results from the realistic simulations indicate that the model does not work very well when no edges are influential. This was to be expected. The model does not handle presence-absence of individual microbes specifically, as this is not relevant to the brain connectome scenario. We'd like to extend the model to specifically handle the presence and absence of these individual microbes in the future. The Julia package is currently available on GitHub and will be submitted to the Julia package distribution soon. If you're interested, we'd love for you to try it out and let us know what you think. Finally, I'd like to thank Claudia, as well as everyone else in the lab. In addition, I'd like to thank Doug Bates and Rick Lankow for all of their help. And finally, thank you to the Department of Energy and the USDA for their support.